an official website of the United States government. Here's how you know. Log in. Access keys: NCPI homepage, My NCPI homepage, main content, main navigation. Preview improvements coming to the PMC website in October 2024. Learn more or try it out now. As a library, NLM provides access to scientific literature. Inclusion in an NLM database does not imply endorsement of or agreement with the contents by NLM or the National Institutes of Health. Learn more. PMC disclaimer, PMC copyright notice. Molecules. 2016 August 21, 8931. Published online 2016 July 27th. DOI 10.3390/molecules 2108091. CID PMC 6274000. PMID 2747238. Capsaicin, from plants to a cancer-suppressing agent. Angela M. Chapo Oliver 1 and Laura Mejia Teniente 2, *. Pinju Cho, academic editor. Author information, article notes, copyright, and license information, PMC disclaimer. Go to. Abstract. Capsaicinoids are plant secondary metabolites, capsaicin being the principal responsible for the pungency of chili peppers. It is biosynthesized through two pathways involved in phenylpropanoid and fatty acid metabolism. Plant capsaicin concentration is mainly affected by genetic, environmental, and crop management factors. However, its synthesis can be enhanced by the use of elicitors. Capsaicin is employed as food additive and in pharmaceutical applications. Additionally, it has been found that capsaicin can act as a cancer preventive agent and shows wide applications against various types of cancer. This review is an approach in contextualizing the use of controlled stress on the plant to increase the content of capsaicin, highlighting its synthesis and its potential use as anti-cancer agent. Keywords: capsaicin, elicitors, stress, cancer, apoptosis, cell death. Go to. 1. Introduction. Pepper is a member of the Solanaceae family and is one of the oldest domesticated crops in the Western Hemisphere. 2. The importance of this crop is that consumption continues to grow because of its high nutritional value, since it is a rich source of vitamin C and E as well as provitamin A and carotenoids, compounds with well-known antioxidant properties. 3. Peppers are mainly consumed as food additives because of their unique pungency, aroma, and color. Only in 2011, the value of global hot pepper production was $14.4 billion, 40-fold higher than in 1980. 1. The genus Capsicum consists of approximately 25 wild and 5 domesticated species. The 5 domesticated species are Capsicum annuum, C. bacatum, C. chinense, C. frutescens, and C. pubescens. Of the domesticated species, C. chinense is the most pungent fruit type. 4. The pungent principles of chili fruit are capsaicinoids, which are alkaloids that accumulate in the placenta of maturing capsicum, chili pepper pods, and have wide applications in food, medicine, and pharmacy. Around 80% to 90% of capsaicinoids present in capsicum consist of capsaicin and dihydrocapsaicin, in a ratio of approximately 1:1 and 2:1, respectively. The rest is comprised of nordihydrocapsaicin, homodihydrocapsaicin, homocapsaicin, norcapsaicin, and nornorcapsaicin. 5, 6, 7. Capsaicin, trans, 8, methyl, N, vanillyl, 6, nonenamide, is a homovanillic acid derivative responsible for the characteristic pungent feeling of the genus Capsicum. Figure 1, 8. It has been found that a change in the acid portion of capsaicin generates analogs with different degrees of pungency. 9. Castillo et al. 10. Obtained 3 analogs of capsaicin, 2 pungent and 1 with very low pungency. To achieve this, they used different acyl chain lengths and chemical substitutes in the aromatic ring. 10. Additionally, this metabolite concentration, like other plant secondary metabolites, can be risen under controlled stress. 11. Both capsaicin and its analogs have been used medicinally for centuries, but recently it has been extensively studied for its analgesic, antioxidant, anti-inflammatory, and anti-obesity properties, and, most recently, Its anti-cancer activity against a variety of cancer types 12, 13, 14, 15, 16, 17, 18. Figure 1. Chemical structure of capsaicin 7. Therefore, the aim of this review is to contextualize the use of controlled stress on the plant to increase the content of secondary metabolites, highlighting the synthesis of capsaicinoids and their potential use as anti-cancer agents. Go to 2. Crops with higher content of capsaicin. The primary capsaicinoids, capsaicin, dihydrocapsaicin, and nordihydrocapsaicin, are produced exclusively in the epidermal cells of the chili placenta. Within the domesticated species of the genus Capsicum, Capsicum chinense is recognized as having the most pungent fruit 19. The pungency level is expressed in Scoville heat units, HUS, which is an arbitrary unit and depends on the palate of the taster. It was developed by Scoville in 1912 and is related to the amount of capsaicin present in the sample. It is known that the human palate can detect it, even in as diluted a ratio as 1 to 17, 000. According to the scale, see Chinense JAQ.CV. Naga King chili, an important pepper crop of India, has been acknowledged as the hottest chili in the world, measuring 1,001,304 SHU. 4. Diverse cultivars of habanero pepper have also occupied the highest values of the Scoville scale. As more pungent cultivars have become known, Red Savina habanero 580,000 SHU, the number of levels in the scale has changed. The C. chinense species continues to occupy the highest places of the scale, 19. Recently, in a comparative study of pungency between the three most pungent cultivars reported, Orange Creole habanero, Red Savina habanero, and the variety Butjalokia, all of which belong to the species Capsicum chinense, Bosland and Burrell 20, found that Butjalokia was significantly more pungent, 1,001,304 SHU, making it the most pungent chili pepper known to date, only surpassed by pure capsaicin, 16 million SHU, whereas the variety Red Savina habanero, despite expectations to the contrary, registered a pungency lower than that of the standard orange habanero pepper, 248,556 versus 357,729 SHU, 19. It is important to note that the Scoville organic test is a subjective measure of chili pungency invented by Scoville in 1912, and since its publication, the cultivars of habanero pepper have occupied the highest values of the scale, 20. Go to 3. Capsaicin biosynthesis. Despite the proposed biosynthetic pathway for capsaicinoid synthesis was presented by Bennett and Kirby, 21, and Leet and Loudon, 22, many enzymes involved in capsaicinoid biosynthesis are not yet well characterized, and regulation of the pathway is not fully understood. It is now known that there are two main routes for the synthesis of capsaicinoids. The first dependent on the route of the phenylpropanoid, where L-phenylalanine derivatives as cinnamic, picoumaric, caffeic and ferulic acid and vanillin lead to formation of vanillylamine for the subsequent condensation of capsaicin through capsaicin synthase. The second route involves the metabolism of branched fatty acids, mainly derived from valine or leucine, ending in the formation of 8-methyl-6-nonenoyl-CoA. Finally, capsaicin is generated through a condensation reaction between vanillylamina and 8-methyl-6-nonenoyl-CoA catalyzed by the coenzyme A-dependent acyltransferase. Figure 2, 23. 24. Figure 2. Capsaicin biosynthetic pathway modified from Aurora et al. 11. Enzymes involved in the biosynthetic pathway: PAL, phenylalanine ammonia lyase, C4H, cinnamic acid 4-hydroxylase, Ca3H, coumaric acid 3-hydroxylase, zone T, caffeic acid O-methyltransferase, PMT, putative aminotransferase, BCT, branched chain amino acid transferase, FAS fatty acid synthase complex, KS, beta-ketoacyl-acyl carrier protein, ACP, synthase, ACL, acyl carrier protein, FATA, acyl ACP thioesterase, denaturase, ACS, acyl-CoA synthase, CS, capsaicin synthase. Significant efforts have been made toward understanding biosynthetic pathway of capsaicin. Examples of this are the genes codified for the enzymes phenylalanine ammoniolase, PAL, cinnamic acid 4-hydroxylase, C4H, caffeic acid O-methyltransferase, COMT, a putative aminotransferase, PMT, and a beta-ketoacyl-acyl carrier protein, synthase, KS, that were identified in the cDNA library obtained from placenta tissue of highly pungent peppers, Capsicum chinense CV habanero, obtained by Curry et al. 7. A key step in capsaicin biosynthesis is the conversion of vanillin to vanillylamine, and a putative aminotransferase, PMT, has been proposed to be the enzyme responsible for vanillin's transamination. Abraham Juarez et al. 24 showed that silencing PMT gene through virus-induced gene silencing, VGS, reduces the level of capsaicin in C. annuamel, CV tampiquino, classified as mildly pungent, thus proving that PMT gene was involved in capsaicinoid synthesis. Additionally, Tanaka et al. 25 showed that a single amino acid substitution in AMT protein sequence led to a functional loss. Several studies have shown that the expression profile of PUN1 gene, coding for a putative capsaicin synthase, CS, correlates with pepper pungency. Ogawa et al. 23, by silencing the PUN1 gene in pepper plants, found that the capsaicin levels in placenta tissues were low, compared with control plants, thus demonstrating that PUN1 gene plays an active role in the synthesis of capsaicin. 23. In addition, it has been reported that PUN1 has a role in the capsinoid synthesis. Capsinoids are capsaicinoid-like substances that lack pungency and possess similar physiological functions to capsaicinoids. They are present in sweet chili varieties, 26, 27, 28. Therefore, they turn out to be promising compounds with similar applications of capsaicin. However, sequencing the chili genome opens a key opportunity to gain a complete and clear understanding of the capsaicinoid pathway and represents an excellent resource for exploring the evolution of secondary metabolites in plants. 1. The study of the hot pepper genome sequence suggests that the pungency has its origin in the evolution of new genes by unequal duplication of existing genes and owing to changes in gene expression in fruits after speciation. 1. Go to 4. Elicitor induction of capsaicinoids. Plants are frequently exposed to different environmental stresses types, both biotic and abiotic. These generate biochemical and metabolic changes, resulting in the production of hydrogen peroxide, H2O2, and reactive oxygen species, ROS. The oxidative burst generated corresponds to the primary response of the defense mechanism in plants, 29, 30. However, this signaling cascade can also be activated by the use of elicitors, stable molecules that induce an immune defense response in plants and accumulation of diverse secondary metabolites as part of the defense responses to pathogen infection and environmental stress. 30. 31. Elicitors can play an important role in the achievement of long-term crop productivity. Through the use of elicitors during growth, phytonutrients content can be maximized while environmental impacts are reduced. 32. Vargas Hernandez et al. 31 confirmed significant increase of secondary metabolites accumulation such as total phenolics, flavonoids, and capsaicinoids contents in variety C. chinense treated with H2O2 compared to control plants. Additionally, they observed an increase in antimicrobial activity in methanolic extracts from plants previously elicited. Effective laminin spray in C. frutescens capsaicinoids levels was tested by Gururaj et al. 33. Their study showed that both vanillylamine, a precursor for all capsaicinoids, and capsaicin levels were 9-fold, 44.57 plus or minus 1.22 mol/g dry weight, and 1.7 folds, 62.65 plus or minus 1.72 mol/g dry weight, higher than levels in control. In addition to the direct effect that the elicitors have on capsaicin accumulation in plants, it has been found that this effect is also achieved in plant cells culture. It was found that root cultures of Capsicum frutescens, treated with veratraldehyde, a derivative of vanillin, accumulated more vanillin, 78 μm, than caffeic acid-fed cultures, 34. Additionally, they observed that efficiencies of biotransformation, respecting to vanillin formation, with caffeic acid and baritraldehyde were 2.2% and 9% respectively, indicating a possible diversion of the phenylpropanoid pathway towards other secondary metabolites. 34. Eilek et al. 35 studied the effect of different cellulase concentrations on the production of capsaicin in freely suspended and immobilized cell cultures of Caramamoras pepper seeds, C. Annuum L. They found that the immobilization process had an increasing effect on the capsaicin accumulation and that the highest capsaicin concentration for the immobilized cells was 362.91 μg/g fresh weight after 24 hours. Go to 5. Anti-cancer activity. There is persuasive epidemiological and experimental evidence that dietary phytochemical found in fruits, vegetables, whole grains, spices, and teas exhibit diverse inhibitory effects against cancer initiation, promotion, progression, and metastasis. 36. Between them is capsaicin, a bioactive phytochemical abundant in chili peppers. Capsaicin is a homovinylic acid derivative, and it has been shown to alter the expression of several genes involved in cancer cell survival, growth arrest, angiogenesis, and metastasis. 17. 18. 37. 38. Tumorigenesis is a multi-stage process that generally occurs over an extended period of time. Cancer cells acquire unique capabilities that most healthy cells do not possess. 18. Cancer is initiated and progresses by multiple genetic alterations and aberrant signaling pathways. Identification of molecular targets involved in the steps of tumor development will provide opportunities to establish a promising strategy to fight against cancer. 18. Studies evaluating the capsaicin effect on cancerous cells are showed in Table 1, where displayed the capsaicin effect to inhibit cell proliferation in many cancer cell types by mechanisms that are not completely understood. 39. The proposed anti-cancer mechanisms of capsaicin include an increase of cell cycle arrest and apoptosis. 18. Table 1. Studies evaluating the capsaicin effect on cancerous cells. Cancer type. Cell line. Effective doses μm. Anti-cancer mechanism. Ref. Human colorectal cancer. HCT 116. 100 to 200. Induced autophagy. 40. LUVO. 100. Induced antitumorigenesis. Deregulation of beta-catenin/TCF-dependent signaling. 41. SW480. 42. Colorado 205. 150. Induced cell death, increased RS and pro-apoptotic proteins. 43. Human breast cancer. MCF 7. 50 to 300. Induced autophagy. Inhibited growth and induced apoptosis. 40. 44. T47D. 200. Inhibited growth, increased apoptosis. 45. 46. 47. 48. 49. 50. BT474. SKBR 3. MDA MB231. 20 to 200. Induced apoptosis and dysfunctions in mitochondria. Antiproliferative activity and arrest of cell cycle into G2/M phase. Enhances the apoptotic effects of trial by activating the calcium, CMPI, SP1 pathway. Human prostate cancer. LNCAP. 40 to 50. Inhibited proliferation. Induced apoptosis. 51, 52, 53, 54, 55. PC3. 20 to 50. DU. 145. 500. RWP1. 40. Human myeloid leukemia. HL. 60. Greater than 50. Induced G0/G1 phase cell cycle arrest and apoptosis. 56. U937. 200. Enhances the apoptotic effects of trial by activating the calcium, CMPI, SP1 pathway. 50. THP1. Human esophageal epidermoid carcinoma. C81T/VGH. 100. Induced apoptosis and G0/G1 phase cell cycle arrest. 44, 49. Human melanoma. A375. 100. Inhibited cell growth and promoted apoptosis. 47, 49. 57. Human KB cancer cells. KB cells. 150 to 250. Reduce cell proliferation and viability. Induce cell death and cell cycle arrest in G2/M phase. 58. Mouse melanoma. B16, F10. 50. Inhibited cell migration. Induced apoptosis. 49, 59. 60. Human pancreatic cancer. As PC1. 150. Inhibited proliferation. Induced apoptosis and generated ROS. 61, 62. BXBC3. PNC1. 200. Induced G0/G1 phase cell cycle arrest and apoptosis. Inhibited growth. 41, 63. Human multiple myeloma. U266. 5. Inhibited cell proliferation caused accumulation of cells in G1 phase. 60. MM.1S. Human hepatoma. HepG2. 10 to 200. Decreased cell viability, generated RS and activated caspase. 3. Induced apoptosis and autophagy. 64, 65. Hep3B. 200. Enhances the apoptotic effects of trial by activating the calcium, CMPI, SP1 pathway. 50. Human nasopharyngeal carcinoma. NPCW039. 200 to 400. Induced G0/G1 phase arrest and apoptosis. Increased RS and activated caspases. Increased cytosolic A2+. 58. Human gastric carcinoma. SMC1. 200. Induced apoptosis. 66. Human bladder cancer. T24. 100. Induced RS production and mitochondrial membrane depolarization. 67. Human small cell lung cancer. NCI, H69. 50. Suppressed growth in all 4 cell lines. 68. NCI, H82. DMS 53. DMS 114. Open in a separate window. Go to 6. Capsaicin and apoptosis. Apoptosis is an essential barrier against cancer development and progression, and loss of apoptotic signaling is highly associated with malignancy. 37. It has been recently demonstrated that capsaicin induces apoptosis in many types of cancer cell lines, including colon adenocarcinoma, pancreatic cancer, hepatocellular carcinoma, prostate cancer, breast cancer, and many others, leaving normal cells unharmed. 17. Nevertheless, the molecular mechanism whereby capsaicin induces apoptosis in cancer cells is not completely elucidated, but involves intracellular calcium increase, RS, disruption of mitochondrial membrane transition potential, and activation of transcription factors such as NF-kappa B and STATs. Figure 3. 69, 70, 71. Figure 3. Summary of some of the pro-apoptotic mechanisms and anti-cancer activity of capsaicin in some cancerous cells. 17, 52, 62, 72, 73, 74. It has been found that pro-apoptotic activity of capsaicin is mediated via TRPV1, transient receptor potential vanilloid type 1, in many types of cancers. 75. TRPV1 is a non-selective cation channel that belongs to the family of transient potential receptors, TRPs. 70. It prefers CA2+ over Na+. Thus, it contributes to changes in cytosolic free, say 2+ concentration, and is the primary cellular target of capsaicin, 75. TRPV1 is also broadly distributed in tissues of the brain, bladder, kidneys, intestines, keratinocytes of epidermis, glial cells, liver and polymorphonuclear granulocytes, mast cells, and macrophages, 76. Until now, capsaicin has been identified uniquely as an agonist of TRPV1. The expression of TRPV1 has been demonstrated in most of the tumor cells analyzed. TRPV1 is expressed in human breast cancer MCF 7 and BT20 cells 45, 77, prostate cancer-derived LNCP and PC3 cells, and benign prostate hyperplasia BPH tissue. Expresses TRPV1 51. Capsaicin has been shown to inhibit growth or cause apoptosis in prostate tumor cells, both in vitro and in vivo. Table 2. TRPV1 expression is upregulated in prostate cancer, but it is not the only vanilloid receptor that is related. It has been found that TRPV6 is upregulated in advanced prostate cancer. Compared with normal tissue or cells, the expression of TRPV6 mRNA and expression of the TRPV6 protein is substantially increased in prostate cancer tissue, 78. Its expression is also regulated by the androgen receptor, which highlights the role of TRPV channels in the growth of prostate cells, 69, 78. Experiments using capsaicin at 10 μm and above resulted in proliferation reduction of the androgen-independent PC3 and DU, 145 cells, and caused apoptosis in vitro, 51. Capsaicin treatment also induced apoptosis in prostate cells by dissipation of the mitochondrial inner transmembrane potential and activation of caspase 351. The same mechanism of action has been observed in human colon, 79, and pancreatic cancer cells, 80, treated with capsaicin. Table 2. Studies evaluating capsaicin anticancer effect in vivo. Animal model. Capsaicin doses. Treatment. Results. Ref. BLB/CJ and BLB/CJ new/on mice injected with live tumor cells CT26. 100 to 200 μg. Intratumoral on days 5, 10, and 15. Retarded progression of injected tumors. 81. BNX new/on male mice injected with PC3 cells. 5 mg per kilogram per day. Gavage 3 days per week for 4 weeks. Reduced tumor growth. 53. Athymic nude mice injected with PC3 cells. 5 mg per kilogram body weight. Subcutaneous injection every 2 days for 14 days. Suppressed PC3 tumor growth and induced apoptosis. 51. Female athymic nude mice injected subcutaneously with ASPC1 tumor cells. 2.5 mg per kilogram body weight. 5 times a week. Suppressed growth of tumor xenografts without adverse effects. 61. 5 mg per kilogram body weight. 3 times a week. 61. Male athymic new mice injected with U266 cells. 1 mg per kilogram. Twice a week for 3 weeks. Inhibited growth of U266 xenograft tumors. 60. Male athymic nude mice injected subcutaneously with T24 cells. 5 mg per kilogram. Subcutaneous injection every 3 days for 4 weeks. Slowed growth of xenograft tumors. 67. Female triple-deficient beige/nude/SID mice, BNX, injected with MDA, MB231 cells. 5 mg per kilogram. Oral gavage 3 days per week for 4 weeks. Decrease the size of tumors by 50%. 46. Male nude mice injected subcutaneously with H69 cells. 10 mg per kilogram body weight. Solid diet until tumors of the control group reached 2000 mm3. Tumor growth suppression. 68. Female BLB/C athymic nude mice injected subcutaneously with Colorado 205 cells. 1 mg per kilogram. Intraperitoneal injected. For weeks of treatment. Inhibition of tumor growth. 46. 3 mg per kilogram. Female athymic nude mice injected subcutaneously with ASPC, 1 tumor cells. 2.5 mg per kilogram. Orally fed 5 days a week for 6 weeks. Reduce tumor SOD activity by 60% and increase the ratio of oxidized glutathione to glutathione. 62. Male BLB/C nu/nu, athymic nude mice injected subcutaneously with PNC, 1 cells. 5 mg per kilogram body weight. Gavage 3 days per week for 4 weeks. Inhibited the growth of pancreatic cancer PNC, 1 cell xenografts. 41. Open in a separate window. Capardossi et al., 75, found that capsaicin treatment induced a more aggressive gene phenotype and invasiveness in null, TRPV1 urothelial cancer cells, 75. However, transfection of TRPV1 cDNA in these cells restores the sensitivity of capsaicin-induced apoptosis and inhibited the acquisition of a more aggressive metastatic phenotype, 75. Additionally, capsaicin-induced apoptosis in low-grade urothelial cancer, but not in high-grade due to the loss of expression of TRV1, 75. On the other hand, studies performed in pancreatic cells showed that capsaicin apoptosis-inducing effects were associated with RS generation, J&K activation, mitochondrial depolarization, release of cytochrome C in the cytosol, and activation of caspase 3 cascade, 82. This result shows that capsaicin is able to activate apoptosis via non-receptor mechanisms, 61, 82. As mentioned above, capsaicin induced RS generation in cancer cells and has been proposed as the principal signaling molecules, 69, 80. Induction of apoptosis in cells is associated with significant elevation of intracellular RS production, 39. The relationship between capsaicin exposure and generation of RS is also quite complex. In normal cells, RS are conventionally considered cytotoxic and mutagenic, and at high levels they can induce cell death, apoptosis, and senescence, 83. It has been suggested that capsaicin induces apoptosis in cancerous cells via the generation of even higher levels of intracellular RS, 83. In recent years, a number of studies have shown that oxidative stress could cause cellular apoptosis via both mitochondria-dependent and mitochondria-independent pathways, Figure 3, 84. In most cells, mitochondria constitute 15% to 50% of the total cytoplasmic volume, and they participate in metabolic functions, especially those involved in cellular energy production, more than any other organelle. In addition, mitochondria consume approximately 90% of cellular oxygen and are the major source of RS, which are generated during respiration and involved in maintaining the intracellular redox state, 80. In addition to their long-standing role in energetics, mitochondria represent a point of convergence for many cell death signals in mammalian cells, 72. Interactions at the mitochondrion ultimately determine whether a cell survives or dies in response to many physiologic or therapeutic cell death stimuli. Capsaicin has been shown to target several proteins involved in the mitochondrial death pathway to initiate apoptosis in different cancer cell lines, 1736. Hale and Lawton, 52, found that a 12H exposure to increasing concentrations of capsaicin was sufficient to promote increasing levels of apoptosis in colo-16 cells. More than half underwent apoptosis, which was associated with progressive dissipation of mitochondrial transmembrane potential, delta-CM, and enhanced superoxide production, reflecting the disintegration of mitochondria and subsequent malfunction of mitochondrial electron transport. NADPH oxidase constitutes part of complex I of the mitochondrial electron transport chain. It has been found that capsaicin directly inhibits mitochondrial NADPH oxidase activity by binding competitively to the ubiquinone/coenzyme Q site on this enzyme, 17. Therefore, if capsaicin blocks electron transport in mitochondria, dissipation of delta-CM should follow. Loss of delta-CM is widely understood to initiate apoptosis through causing mitochondrial permeability, which leads to the release of cytochrome C and subsequent activation of pro-apoptotic pathways. 52. A recent study has shown the involvement of mitochondrial electron transport chain (ETC) complexes I and III in capsaicin-induced apoptosis. Pramanik et al. 62 evaluated the mechanism of capsaicin-mediated RS generation in pancreatic cancer cells. They found that capsaicin reduces complex I and complex III activity in BXPC3 and SPC1 cells, leading to RS generation. Additionally, they also found that the antioxidant levels were lower in capsaicin-treated mice tumor cells compared to control, resulting in the accumulation of RS and mitochondrial damage. 62. Therefore, capsaicin treatment generates RS through mitochondria and lowers intracellular antioxidants levels, resulting in mitochondrial damage and apoptosis in pancreatic cancer cells. 62. Capsaicin can also inhibit the plasma membrane NADH oxidase by functioning as a coenzyme Q antagonist. The vanylyl moiety of capsaicin is structurally similar to the cyclic portion of coenzyme Q, which could account for the fact that vanilloids act as coenzyme Q antagonists. The inhibition of the plasma membrane NADH oxidase has been reported to be associated with the pro-oxidant and pro-apoptotic properties of capsaicin in certain transformed cells and activated T cells. 73. AMPK interaction is another mechanism proposed by the anti-cancer activity of capsaicin. AMP-dependent protein kinase, AMPK, considered the principal metabolic gatekeeper of the cell, is a member of a protein kinase family that is activated during ADP-depleting metabolic states, such as hypoxia, heat shock, oxidative stress, and exercise. 62. It functions as a major metabolic switch to maintain energy homeostasis and has been shown to exert as an intrinsic regulator of mammalian cell cycle. 85. 86. It is activated by metabolic stresses that increase cellular ADP/ATP and/or AMP/ATP ratios. 87. Activation of AMPK has been related to apoptosis due to metabolic stress and, therefore, AMPK has been proposed as an apoptotic molecule. AMPK activation induced apoptosis in many human cancer cells and enhanced oxidative stress during apoptosis. 85. 86. Upon energetic imbalance, intracellular concentrations of AMP increase, thus promoting AMPK activation. Activated AMPK stimulates catabolic pathways and, concomitantly, inhibits the rate of anabolic reactions to restore the correct adenylate energy charge. It has been shown that capsaicin treatment of colon cancer HT29 cells induced AMPK activation and inhibition of ACC, which is a well-known AMPK substrate, suggesting that capsaicin inhibits lipid biosynthesis. Both phenomena were involved in capsaicin-induced apoptosis. 86. Go to 7. Capsaicin, cell cycle, and P53. Cells proliferate through the cell cycle, which is divided into G0/G1, S, and G2/M phases. Through the cell cycle, DNA checkpoints exist to ensure the integrity of DNA replication. These checkpoints and repair pathways facilitate cellular responses to DNA damage. 36. Any alteration in these pathways can increase the risk of cancer. Essential parts of the cell cycle machinery are the cyclins, cyclin-dependent kinases, CDKs, and the CDK inhibitors. Once activated, the CDKs provide a driving force for the cells to move from one phase to the next, but if cyclin and/or CDKs are affected, cell cycle arrest occurs. 39. 87. 88. 89. Chen et al. 88. Found that capsaicin inhibits the proliferation of 5,637 bladder carcinoma cells by cycle arrest with the inhibition of CDK2, CDK4, and been shown to play an important role in a number of inflammatory diseases. 39. Several studies have demonstrated the tumor cytokines and chemokines have been shown to play an important role in a number of inflammatory diseases. 39. Several studies have demonstrated that RS generation plays a significant role in phosphorylation of p53 at the SER15 residue, 89. Following capsaicin treatment, an increase in p53 and phosphorylated p53 has been observed. The tumor Go to 8. Conclusions. Capsaicinoids, and particularly capsaicin, are bioactive compounds exhibiting characteristics of great interest to researchers, including for their pharmacobiology applications, such as their recent applications against cancer. Although various capsicum species contain high amounts of capsaicin, not all of these species are used as a source of capsaicin. Cytokines and chemokines have been shown to play an important role in a number of inflammatory. The tumor suppressor protein p53 regulates the cellular response to DNA damage by mediating cell cycle arrest, DNA repair, and cell death. Phosphorylation at the SER15 residue of p53 is critical for p53-dependent transactivation, 39. Capsaicin was found to induce p53 phosphorylation at the SER15 residue and enhanced p53 acetylation through downregulation of sirtuin-1, which is responsible for activation of apoptosis, 74. Experiments suggest that p53 is a target of the anti-cancer mechanism of capsaicin. Arnab et al., 70, incubated human gastric cancer AGS cells with various concentrations of capsaicin in the presence and absence of p53 sirna. They found that capsaicin induces apoptosis in AGS cells through upregulation of p53 and that the apoptotic activity of capsaicin is p53 dependent. They also found that the ability of capsaicin to induce the expression of pro-apoptotic proteins such as Bax, caspase-3, and caspase-8 was almost completely obliterated by knocking down p53, 70. Park et al., 90. Investigated the effects of capsaicin in the same type of cells and found that the activity of caspase-3 increased with the exposure to capsaicin, suggesting that capsaicin may serve as an antitumorigenic agent in human gastric cancer. Cytokines and chemokines have been shown to play an important role in a number of inflammatory diseases, 39. Several studies have demonstrated that RS generation plays a significant role in phosphorylation of p53 at the SER15 residue, 89. Following capsaicin treatment, an increase in p53 and phosphorylated p53 has been observed in several cancer cell lines, 57, 77, 90, 91, 92. Go to 8. Conclusions. Capsaicinoids, and particularly capsaicin, are bioactive compounds exhibiting characteristics of great interest to researchers, including for their pharmacobiology applications, such as their recent applications against cancer. Although various capsicum species contain high amounts of capsaicin, not all of these species are used as a source of capsaicin. Therefore, new strategies have been used to increase the content of this secondary metabolite and thereby generate lines of research that lead from crop care and harvesting to capsaicin purification and application in cancer cell lines. One of these is the use of elicitors, both in crops and plant cell cultures. Further study of the anti-cancer targets of capsaicin holds potential for novel therapies in the future and warrants more research to improve our understanding of its efficacy in cancer prevention and treatment. Go to acknowledgements. We thank Universidad de Guanajuato and CONACYT Mexico for supporting this work. The authors also thank Ramón Gerardo Guevara González and Ireneo Torres Pacheco, researchers from Universidad Autónoma de Querétaro, for their partial support to carry out this work. Go to author contributions. All authors contributed equally to the realization of this article. DRA. Angela M. Chapa Oliver reviewed the issue of cell lines and summarized the molecular anti-cancer role of capsaicin. DRA. Laura Mejia reviewed the issue of capsaicin increased with the use of elicitors and key points of the biosynthetic pathway of capsaicin. Go to conflicts of interest. The authors declare no conflict of interest. Go to references. 1. Kim S., Park M., Yun S.I., Kim Y.M., Lee J.M., Seo E., Choi J., Chong K., Kim K., T., Young K., et al. Genome sequence of the hot pepper provides insights into the evolution of pungency in capsicum species. Not Janet. 2014. 46:270, 279. doi 10.1038/ing.2877. PubMed, Crossref, Google, Scholar. 2. Aguilar Melendez A., Morel P.L., Roos M.L., Kim S.C. Genetic Diversity and Structure in Semi-Wild and Domesticated Childs, Capsicum annuum, Solanaceae, from Mexico. A.M. J. Bot 2009, 96:1190, 1202. doi 10.3732/AJ.0800155. PubMed, Crossref, Google Scholar. 3. Mater Ska M. Paruca I. Antioxidant activity of the main phenolic compounds isolated from hot pepper fruit, Capsicum annuum L. J. Agric, Food Chemistry 2005, 53:1750, 1756. doi 10.1021/JF035331K. PubMed, Crossref, Google Scholar. 4. Kihi M. Camaria S. Tandon P. Manipulation of culture strategies to enhance capsaicin biosynthesis in suspension and immobilized cell cultures of Capsicum chinense Jack. CV Naga King Chili. Bioprocess Biosist. ENG 2014, 37:1055, 1063. doi 10.1007/S00449-013-1076-2. PubMed, Crossref, Google Scholar. 5. Govindarajan vs. Satyanarayana MN Capsicum Production, Technology, Chemistry, and Quality. Part 5. Impact on Physiology, Pharmacology, Nutrition and Metabolism, Structure, Pungency, Pain, and Desensitization Sequences. Crit. Reverend Futsai, Noter. 1991, 29:435, 473. doi 10.1080/104083991095275536. PubMed, Crossref, Google Scholar. 6. Prasad NBC, Shrivastava, Ravi Shankar Ga capsaicin as multifaceted drug from Capsicum SPP. Evid. Based Med, 2005, 2:147, 166. doi 10.2165/0117065-200502030-00006. Crossref, Google Scholar. 7. Curry J. Aluru M. Mendoza M. Navarez J. Melendrez M. O'Connell M.A. Transcripts for possible capsaicinoid biosynthetic genes are differentially accumulated in pungent and non-pungent capsicum. Plant Sci. 1999, 148:47, 57. doi 10.1016/S0168-9452, 99, 00118-1. Crossref, Google Scholar. 8. European Commission Health and Consumer Protection Directorate General. Opinion of the Scientific Committee on Food on Capsaicin. European Commission Health and Consumer Protection Directorate General, Brussels, Belgium, 2002. Pages 1-12. Google Scholar. 9. Al-Snafi A. The pharmacological importance of capsicum species, Capsicum annuum and Capsicum frutescens, grown in Iraq. J. Farm. Biol. 2015, 5:124, 142. Google Scholar. 10. Castillo E. Lopez Gonzalez I. De Regil Hernandez R. Reyes Duarte D. Sanchez Herrera D. Lopez Munguia A. Darson A. Enzymatic synthesis of capsaicin analogs and their effect on the T-type CA2+ channels. Biochem. Biophys. Res Com 2007, 356:424, 430. doi 10.1016/Jade. BBRC.2007.02.144. PubMed, Crossref, Google Scholar. 11. Aurora R. Gil N.S., Chauhan G., Rana A.C. An overview about versatile molecule capsaicin. Int J. Pharm. Sci. Drug Res 2011. 3:280, 286. Google Scholar. 12. Simone D.A., Baumann T.K., Lamott R.H. Dose-dependent pain and mechanical hyperalgesia in humans after intradermal injection of capsaicin. Pain. 1989, 38:99, 107. doi. 10.1016/0304-3959, 89, 90079-1. PubMed, Crossref, Google. Scholar. 13. Brederson J.D., Kim P.R., Selassie A. Targeting TRP channels for pain relief. Year J. Pharmacol. 2013. 716:61, 76. doi. 10.1016/j.edgevar.2013.03.003. PubMed, Crossref, Google Scholar. 14. Galano A., Martinez A., capsaicin, a tasty free radical scavenger, mechanism of action and kinetics. J. Phys. Chemistry 2012, 116:1200, 1208. doi. 10.1021/JP211172F. PubMed, Crossref, Google Scholar. 15. Kim C.S., Kawada T., Kim B.S., Han I.S., Cho S.Y. Capsaicin exhibits anti-inflammatory property by inhibiting ICBA degradation in LPS-stimulated peritoneal macrophages. Cell Signal. 2003, 15:299, 306. doi. 10.1016/S0898-6568, 02, 00086-4. PubMed, Crossref, Google Scholar. 16. Kong J.H., Kim C.S., Han I.S., Kawada T. U.R., capsaicin, a spicy component of hot peppers, modulates adipokine gene expression and protein release from obese mouse adipose tissues and isolated adipocytes, and suppresses the inflammatory responses of adipose tissue macrophages. F. Bieslet. 2007, 581:4389, 4396. doi. 10.1016/j.febslit.2007.07.082. PubMed, Crossref, Google Scholar. 17. Blake K.B., Borman G., Muhammad B., Mackenzie D., Faber S. A comprehensive review of the carcinogenic and anticarcinogenic potential of capsaicin. Toxicol. Pat Hall. 2012, 40:847, 873. doi. 10.1177/019262331244441. PubMed, Crossref, Google Scholar. 18. Clark R., Lee S., Anti-Cancer Properties of Capsaicin Against Human Cancer. Anti-Cancer Res 2016, 36:837, 844. PubMed, Google Scholar. 19. Contoflick A., Thalamu C.E., Bello J.J., Lacona Guzman C., Solis Marroquin D., Avilas Venus S., Gomez U.C.E., Lopez P.U.C.G., Santana Buzzy N., capsaicinoids content in habanero pepper, Capsicum chinense Jack, hottest known cultivars. Hort Science. 2008, 43:1344, 1349. Google Scholar. 20. Bosland P.W., Baral J.B. Butjalokia. The world's hottest known chili pepper is a putative naturally occurring interspecific hybrid. Hort Science. 2007, 42:222, 224. Google Scholar. 21. EYK, Suzuki T., Fujiwake H., Formation and accumulation of pungent principle of hot pepper fruits, capsaicin, and its analogues in Capsicum annuum var. Annuum CV Cariot subasa at different growth stages after flowering. Agric. Biol. Chemistry 1979, 43:2493, 2498. Google Scholar. 22. Leet E. Loudon MCL Biosynthesis of capsaicin and dihydrocapsaicin in Capsicum frutescens. J. A.M. Chemistry SOC 1968, 90:6837, 6841. doi 10.1021/JAWS01026A049. PubMed, Crossref, Google Scholar. 23. Ogawa K, Murota K, Shimura H, Furuya M, Tagawa Y, Matsumura T, Masuda C. Evidence of capsaicin synthase activity of the PUN1 encoded protein and its role as a determinant of capsaicinoid accumulation in pepper. BMC Plant Biol, 2015, 15:1, 10. doi 10.1186/S12870-015-0476-7. PMC free article, PubMed, Crossref, Google Scholar. 24. Abraham Juarez M.A., Rocha Granados M.C., Lopez M.G., Rivera Bustamante R.F., Ochoa Alejo N. Virus-induced silencing of COMT, PMT and Cas genes results in a reduction of capsaicinoid accumulation in chili pepper fruits. Planta. 2008, 227:681, 695. doi 10.1007/S00425-007-0651-7. PubMed, Crossref, Google Scholar. 25. Tanaka Y, Hosokawa M, Miwa T, Watanabe T, Yazawa S. Newly mutated putative aminotransferase in non-pungent pepper, Capsicum annuum, results in biosynthesis of capsinoids, capsaicinoid analogs. J. Agric, Food Chemistry 2010, 58:1761, 1767. doi 10.1021/JF903282R. PubMed, Crossref, Google Scholar. 26. Qin C, Yu C, Shin Y, Fong X, Chen L, Min J, Chung J, Zhao S, Su M, Lua Y, et al. Whole genome sequencing of cultivated and wild peppers provides insights into capsicum domestication and specialization. Proc. Nat. Academic. Sci. USA. 2014. 111:5135, 5140. doi 10.1073/NOS.14009751111. PMC free article, PubMed, Crossref, Google Scholar. 27. Stewart C, Mazarek M, Stellari G M, O'Connell M, John M. Genetic control of pungency in C. Chinense via the PUN1 locus. J. EXP Bot 2007, 58:979, 991. doi 10.1093/JXB/EARL243. PubMed, Crossref, Google Scholar. 28. Topaz A, Ozdemir F. Assessment of carotenoids, capsaicinoids, and ascorbic acid composition of some selected pepper cultivars, Capsicum annuum L. Grown in Turkey. J. Food Compass. Anal. 2007. 20:596, 602. doi 10.1016/Jade, Jivka.2007.03.007. Crossref, Google Scholar. 29. Mejiatinian L, Torres Pacheco I, Gonzalez Shavira MM, Ocampo Velasquez RV, Herrera Ruiz G, Chapo Oliver AM, Guevara Gonzalez RG. Use of elicitors as an approach for sustainable agriculture. Africa J, Biotechnol. 2010, 9:9155, 9162. Google Scholar. 30. Mejiatinian L, Duran-Flores FD, Chapo Oliver AM, Torres Pacheco I, Cruz Hernandez A, Gonzalez Shavira MM, Ocampo Velasquez RV, Guevara Gonzalez RG. Oxidative and molecular responses in Capsicum annuum L after hydrogen peroxide, salicylic acid, and chitosan foliar applications. Int J. Mol Sci. 2013, 14:10178, 10196. doi 10.3390/IGMS140510178. PMC free article, PubMed, Crossref, Google Scholar. 31. Vargas Hernandez M, Torres Pacheco I, Gautier F, Alvarez Mayorga B, Cruz Hernandez A, Garcia Meyer L., Jimenez Garcia SN, Ocampo Velasquez RV, Ferragrino Perez A. Guevara Gonzalez RG. Influence of hydrogen peroxide foliar applications on in vitro antimicrobial activity in capsicum chinense Jack. Plant biocist. Int J. Deal. ASP. Plant Biol. 2016 doi. 10.1080/1126350.2016.11684494. Crossref. Google Scholar. 32. Garcia Meyer L., Guevara Gonzalez RG, Mondragonolbuin VM, Delverduz Coque RBR, Torres Pacheco I. Agriculture and Bioactives, Achieving Both Crop Yield and Phytochemicals. Int J. Mol Sci. 2013. 14:4203, 4222. doi. 10.3390/IJMS140242003. PMC free article, PubMed, Crossref. Google Scholar. 33. Gururaj A.B., Giridhar P., Ravi Shankar G.A. Laminarin as a potential non-conventional elicitor for enhancement of capsaicinoid metabolites. Asian J. Plant Sci. Res 2012. 2:490, 495. Google Scholar. 34. Suresh B., Ravi Shankar G.A. methyl jasmonate modulated biotransformation of phenylpropanoid-sto-vanillin-related metabolites using capsicum frutescens root cultures. Plant Physio. Biochem. 2005. 43:125, 131. doi. 10.1016/J.Plaffy.2005.01.006. PubMed, Crossref. Google Scholar. 35. Ilexi, Astaneus, Coach E. The effects of cellulase on capsaicin production in freely suspended cells and immobilized cell cultures of Capsicum annuum L. Pac. J. Bot 2014. 46:1883, 1887. Google Scholar. 36. Agawal BB molecular targets of dietary agents for prevention and therapy of cancer. Biochem. Pharm. 2006. 71:1397, 1421. doi. 10.1016/J. BCP.2006.02.009. PubMed, Crossref, Google Scholar. 37. Hanahan D. Weinberg R.A. Hallmarks of Cancer, The Next Generation. Cell. 2011. 144:646, 674. doi. 10.1016/J. Cell.2011.02.013. PubMed, Crossref, Google Scholar. 38. Johnson W. Junior Final Report on the Safety Assessment of Capsicum annuum Extract, Capsicum annuum Fruit Extract, Capsicum annuum Resin, Capsicum annuum Fruit Powder, Capsicum frutescens Fruit, Capsicum frutescens Fruit Extract, Capsicum frutescens Resin, and Capsaicin. Int J. Toxicol. 2007. 26:3, 106. doi. 10.1080/10915810601163939. PubMed, Crossref. Google Scholar. 39. Oyagbemi A. Saba A.B., Aziz Oi capsaicin, a novel chemopreventive molecule and its underlying molecular mechanisms of action. Indian J. Cancer. 2010. 47:53, 58. doi. 10.4103/0019-509x.58860. 40. OSH, Kim YS, Lim SC, Ho YF, Cheng IY, UAJ dihydrocapsaicin, DHC, a saturated structural analog of capsaicin, induces autophagy in human cancer cells in a catalase-regulated manner. Autophagy. 2008. 4:1009, 1019. 41. Yang J, Luo B, Su G, Li T, Chen Y, Zhang T. Low concentration capsaicin promotes colorectal cancer metastasis by triggering RS production and modulating ACT/mTR and STAT3 pathways. Neoplasma. 2013. 60:364. 42. Lee S.H., Richardson R.L., Dashwood R.H., Beck S.J. Capsaicin represses transcriptional activity of beta-catenin in human colorectal cancer cells. J. Nutter. Biochem. 2012. 43. Lu H.F., Chen Y.L., Yang J.S., Yang Y.Y., Lu J.Y., Su S.C., Lai K.C., Chung J.G. Antitumor activity of capsaicin on human colon cancer cells in vitro and Colorado 205 tumor xenografts in vivo. J. Agric. Food Chemistry 2010. 58:1. 44. Wu C.C., Lin J.P., Yang J.S., Cho S.D., Chen S.C., Lin Whitey, Lin H.L. Chung J.G. Capsaicin-induced cell cycle arrest and apoptosis in human esophagus epidermoid carcinoma CE81T/VGH cells through the elevation of intracellular reactive oxygen species and Ca2+ productions and caspase 3 activation. Mutat. Res 2006. 45. Cheng H.C., Chen S.D., Qian S.Y., Kwa S.J., Sai H.T., Chin Dr. Capsaicin may induce breast cancer cell death through apoptosis-inducing factor involving mitochondrial dysfunction. EXP Toxicol. 2011. 46. Thenison N.H., O'Kelly J., Lu D., Iwansky G.B., Law D.T., Abbasi S., Leiter A., Carlin B., Meta R., Kofler H.P. Capsaicin causes cell cycle arrest and apoptosis in ER-positive and negative breast cancer cells by modulating the EGFR/HER2 pathway. Oncogene. 2010. 47. Do D. Ahmad A. Yang H. Sarkar FH tumor cell growth inhibition is correlated with levels of capsaicin present in hot peppers. Nutter. Cancer. 2011. 48. Desaw Jr. PL, Pasqualotto KF, Ferreira AK, Tavares MT, Damiao MC, De Azevedo RA, Camara DD, Pereira A, De Souza DM, Filio RPRPF 101, a new capsaicin-like analog, disrupts the microtubule network accompanied by arrest in the G2/M phase, inducing apoptosis and mitotic catastrophe in the MCF7 breast cancer cells. Toxicol. APPL. Pharmacol. 2013. 249. IPSW. Lon SH. Lu HF, Huang AC, Yang JS, Lin JP, Huang HY, Lin JC, Ho CC, Xiu CF. Et al. Capsaicin mediates apoptosis in human nasopharyngeal carcinoma NPC W039 cells through mitochondrial depolarization and endoplasmic reticulum stress. EXP Toxicol. 2011. 50. Moondio, Kong CH, Kong SH, Choi YH, Hyun JW, Chong WY, Kong H, K, Ko Y, S, Meng Y, H, Kim Y, R, et al. Capsaicin sensitizes trial-induced apoptosis through SP1. Mediated DR5 upregulation, involvement of Ca2+ influx. Toxicol. APPL. Pharmacol. 2012. 51. Sanchez AM, Sanchez MG, Malagari Kaznav S, Olia N, Diaz Laviata I, induction of apoptosis in prostate tumor PC3 cells and inhibition of xenograft prostate tumor growth by the vanilloid capsaicin. Apoptosis. 2006. 52. Hale N, Lawton R, examining the role of mitochondrial respiration in vanilloid-induced apoptosis. J. Napple. Cancer INST 2004. 53. Mori A, Lehman S, O'Kelly J, Kumagai T, Desmond JC, Pervin M, McBride WH, Kazaki M, Kofler HP capsaicin, a component of red peppers, inhibits the growth of androgen-independent p53 mutant prostate cancer cells. Cancer Res 2006. 54. Sanchez AM, Martinez Botis J, Malagari Kaznav S, Olia N, Vara D, L'Assumption MA, Diaz Laviata I. Induction of the endoplasmic reticulum stress protein GAD153/CHOP by capsaicin in prostate PC3 cells, a microarray study. Biochem. Biophys. Russ Commune. 2008. 55. Sanchez AM, Malagari Kaznav S, Olea N, Vara D, Kilox A, Diaz laviata I. Apoptosis induced by capsaicin in prostate PC3 cells involves ceramide accumulation, neutral sphingomyelinase, and JNK activation. Apoptosis. 2007. 56. Sao MF, Lu HF, Chin SC, Wu LT, Chin YS, Qua HM, Lin SS, Chang JG involvement of BAX, BCL, 2, CA2+, and caspase, 3 in capsaicin-induced apoptosis of human leukemia HL, 60 cells. Anti-cancer Res 2006. 57. Kim My nitric oxide triggers apoptosis in A375 human melanoma cells treated with capsaicin and resveratrol. Molmed, Republic 2012, 5:585, 591. doi 10.3892/MMR.2011.688. PubMed, Crossref, Google Scholar. 58. Lin CH, Lu WC, Wang CW, Chan YC, Chin MK capsaicin induces cell cycle arrest and apoptosis in human KB cancer cells. BMC complement. Altern. Med. 2013. 59. June HS, Park T, Lee CK, Kong MK, Park MS, Kong HI, Sir YJ, Kim OH capsaicin induced apoptosis of B16, F10 melanoma cells through downregulation of BCL. 2. Food chemistry toxicol. 2007. 60. Bhutani M, Padak AK, Nair AS, Kanamakara AB, Guha S, Sethi G. Agarwal BB capsaicin is a novel blocker of constitutive and interleukin 6 inducible STAT3 activation. Clin. Cancer Res 2007. 61. Zhang R, Humphreys I, Sahu RP, Sher Y. Shrivastava SK. In vitro and in vivo induction of apoptosis by capsaicin in pancreatic cancer cells is mediated through RS generation and mitochondrial death pathway. Apoptosis. 2008. 62. Pramanik KC, Bordi SR, Shrivastava SK role of mitochondrial electron transport chain complexes in capsaicin-mediated oxidative stress leading to apoptosis in pancreatic cancer cells. Pilo S1. 2011. 63. Zhang JH, Lai FJ, Chin H, Luo J, Zhang RY, Bu HU, Wang Z, H, Lin H, Lin S, Z. Involvement of the phosphonocetide 3 kinase/ACT pathway in apoptosis induced by capsaicin in the human pancreatic cancer cell line PNC. 1. OnCall. Let. 2013. 64. Wang SB, Chin JC, Wu CC, Chin CT, Tang New York, Ho Whitey, Lo C, Lin JP, Chung JG, Lin JG capsaicin-induced apoptosis in human hepatoma Hep G2 cells. Anti-cancer Res 2009. 65. Chin X, Tan M, Fong B, Zhao Z, Yang K, Hu C, Liao N, Wang T, Chin D, Xia F, et al. Inhibiting RS, STAT3, dependent autophagy enhanced capsaicin. Induced apoptosis in human hepatocellular carcinoma cells. Free Radik. Res 2016. 66. Wang HM, Chu PJ, Cheng SB, Yang CL, Xiaocaine effect of capsaicin on TNX, in NOX2, protein expression in stomach cancer cells. Biofactors. 2009. 67. Yang ZH, Wang XH, Wang HP, Hu LQ, Zheng XM, Li SW capsaicin mediates cell death in bladder cancer T24 cells through reactive oxygen species production and mitochondrial depolarization. Urology. 2010. 68. Brown KC, Witt TR, Hardman WE, Luo H, Chin YC, Carpenter AB, Lao JK, Dasgupta P. Capsaicin displays antiproliferative activity against human small cell lung cancer in cell culture and nude mice models via the E2F pathway. Pilo S1. 2010. 69. Diaz-Levada I, effective capsaicin on prostate cancer cells. Future OnCall. 2010. 70. Arnav S, Bhattacharjee S, Mundell DP induction of apoptosis by eugenol and capsaicin in human gastric cancer AGS cells elucidating the role of p53. Asian Pac. J. Cancer Previous. 2015. 71. Shin DH. Kim OH. June HS. Kong MK inhibitory effect of capsaicin on B16, F10 melanoma cell migration via the phosphatidylinositol 3 kinase/ACT, RAC1 signal pathway. EXP Mol Med. 2008. 72. Bola PD. Letalier. Mitochondria. Judges and executioners of cell death sentences. Mol Cell. 2016. 61:690. 73. Sur YJ. More than spice, capsaicin in hot chili peppers makes tumor cells commit suicide. J. Natal. Cancer INST 2002. 94. 74. Ito K. Nakazato T. Yamato K. Miyakawa Y. Yamada T. Hozumi N. Sagawa K. Ikeda Y. Kazaki M. Induction of apoptosis in leukemic cells by homovinylic acid derivative, capsaicin, through oxidative stress, implication of phosphorylation of p53 at SER15 residue by reactive oxygen species. Cancer Res 2004. 64. 75. Capridossi S. Amantini C. Nabissi M. Morelli B. Farfariello V. Santilli M. Gismondi A. Santilli G. Capsaicin promotes a more aggressive gene expression phenotype and invasiveness in null TRPV1 urothelial cancer cells. Carcinogenesis. 2011. 76. Reyes Escogido ML. Gonzalez Mondragon E.G. Vasquez Sampansi E. Chemical and pharmacological aspects of capsaicin. Molecules. 2011. 77. Vercelli C. Barbero R. Cuniberti B. Odor R. Re-G. Expression and functionality of TRPV1 receptor in human MCF7 and canine CF.41 cells. Vet. Comp. OnCall. 2013. 78. Pravarskia N. Zhang L. Burrit G. TRP channels in cancer. Biochim. Biophys. ACTA. 2007. 79. Yang Kam. Pyo Jo. Kim Juai. UR. Han Is. Ju SA. Kim WH. Kim VS capsaicin induces apoptosis by generating reactive oxygen species and disrupting mitochondrial transmembrane potential in human colon cancer cell lines. Cell. Mol Biol. Let. 2009. 80. Dutta P. Pramanik Casey. Merotra S. Srivastava SK capsaicin-mediated oxidative stress in pancreatic cancer. In. Prieti V. Editor. Cancer, Oxidative Stress and Dietary Antioxidants. First edition Elsevier, London, UK. 2014. 81. Beltran J. Ghosh AK. Basse S. Immunotherapy of tumors with neuroimmune ligand capsaicin. J. Immunol. 2007. 178. 82. Athanasiu A. Smith PA. Bakalpour S. Kumaran NM. Turner A. Bagioku D. Layfield R. Ray D. Westwell AD. Alexander SPH. Et al. Vanilloid receptor agonists and antagonists are mitochondrial inhibitors. How vanilloids cause non-vanilloid receptor-mediated cell death. Biochem. Biophys. Res Commun. 2007. 83. Hale N. Lawton R. Cancer chemoprevention and mitochondria, targeting apoptosis in transformed cells via the disruption of mitochondrial bioenergetics/redox state. Mol Nutter. Food Res 2009. 84. Sinha K. Das J. Pal PB. Sil PC oxidative stress, the mitochondria-dependent and mitochondria-independent pathways of apoptosis. Archtoxicol. 2013. 85. Kahada FP AMP-activated protein kinase and human cancer, cancer metabolism revisited. Int J. Obes. 2008. 86. Kim YM, Huang JT, Kwok DW, Li YK, Park OJ involvement of AMPK signaling cascade in capsaicin-induced apoptosis of HT29 colon cancer cells. N. Y. Academic. Sci. 2007. 87. Hardy DG, Alessi Dr. ALK B1 and AMPK, and the cancer metabolism link 10 years after. BMC Biol. 2013. 11:88. Chen D. Yang Z. Wang Y. Zhu Ji. Wang X. Capsaicin induces cycle arrest by inhibiting cyclin-dependent kinase in bladder carcinoma cells. Int J. Ural. 2012. 19:89. Mighty R. Sharma J. Jana NR. Capsaicin induces apoptosis through ubiquitin proteasome system dysfunction. J. Cell Biochem. 2010. 90. Park SY. Kim JY. Lee SM. Jun CH. Cho SB. Park CH. Ju YE. Kim SH. Choi SK. Capsaicin induces apoptosis and modulates MAPK signaling in human gastric cancer cells. Mol Med. Republic 2014. 91. Yoon JH on SG. Lee BH. Young SH. OSH role of autophagy in chemoresistance. Regulation of the ATM-mediated DNA damage signaling pathway through activation of DNA-PKCS and PRP1. Biochem. Pharmacol. 2012. 92. Cho CC Wu YC. Wang YF. Cho MJ. Kwa SJ. Chin DR. Capsaicin-induced apoptosis in human breast cancer, NCF. 7 cells through caspase-independent pathway. OnCall. Republic 2009. 21:665. 671. PubMed. Google Scholar. Articles from molecules are provided here courtesy of Multidisciplinary Digital Publishing Institute, MDPI. AMA APA MLA NLM. Follow NCPI. Connect with NLM. National Library of Medicine. 8600 Rockville Pike. Bethesda, Maryland. 20894. Web Policies. FOIA. HHS Vulnerability Disclosure. Help. Accessibility. Careers. NLM. NIH. HHS. USA.gov.